All right, the next chapter that we're going to cover is Chapter 20, and this covers recombinant technologies. In this chapter, by the end of it, you should be able to illustrate how DNA can be manipulated using laboratory techniques, design experiments to construct recombinant DNA molecules, understand techniques used to clone, sequence, analyze, or amplify DNA, and apply DNA technology to modified biological systems. So now we're really going into the realm of biotechnology and experiment. So if you go into a laboratory, if you go into a genetics laboratory or a molecular biology laboratory, or really many different biology laboratories, will all use these techniques. And the crux of this is that we've hijacked biological systems to start to do things that we want to do. And this is kind of how it works. So even nowadays, if you wanted to try to make a lot of money, one of the things you might try to do is isolate a new protein that does something very cool, very unique, and figure out how you can use this to apply in order to do things like create DNA that you want, modify organisms that you want. One of the most recent examples of this is CRISPR-Cas9. So CRISPR-Cas9 came from the fundamental study of bacterial phage resistance, so how bacteria actually resist phage infections, and now that has been turned into a technology that hopefully will allow us to edit any sort of organism in any way that we choose. So now we can think about fixing genetic mistakes that might be in our genome, or if we wanted to do some sort of experiment in a mouse or in a cichlid fish, or in any sort of different model organism, we can start to think about how we want to manipulate DNA to do those experiments. And those are all just because we performed a lot of fundamental biological research, and we can now apply that to these new fields. And so all of this chapter is just kind of how we go about doing that. And so we're going to focus on recombinant DNA technology. So these are technologies that will allow us to mix and match gene sequences, DNA sequences of interest. And so the toolkits, so these are the proteins that have been discovered, the enzymes that have been discovered that we now use on a regular basis in the laboratory. So we can order these things. We just go to NEB or some other company, and we just look up some of these that we want, and we can order it, and within about 3 days you'll get a little tube that contains these. So these include things like restriction enzymes, ligases. So we've covered ligases in the previous chapter on DNA replication, polymerases. Again, these were isolated from fundamental studies of DNA replication, recombinases. We also use things like dyes and radioactive probes. So these are not things that are genetic in basis. These are not built out of DNA. These are not built out of protein. These are other sort of chemical molecules, but they often have some sort of useful and interesting features, such as they are fluorescent, so they give off light, or they can absorb light and emit it in a different wavelength. DNA binding, so dyes that can actually incorporate into DNA, so we can actually mark the presence of DNA within a sample. pH-sensitive dyes, so dyes that change color when pH changes. And then there's also molecular chemistries that we like to use. So nucleotides, so we can simply order these nucleotides, and we have a source of DNA replication. We can order primers. So if you want a short piece of DNA sequence, you can order that, and you can specify it. I want it to be ACC GGT, and then vectors. So these are plasmids that we use in order to grow bacteria that contains DNA sequences that we want. And so we'll go over a lot of this. So this chapter actually covers a lot of different topics, a total of 6 other topics. So recombinant DNA, cDNA libraries, PCR. So we covered that in the previous activity a little bit. Blots, DNA sequencing, and then we're going to get into how you actually engineer an organism, focusing on mouse.